Hello, Marcia.
Hello, hi, my name is Marcia Santos. I am a staff application scientist at SCIEX Operations, and I'm here in beautiful Amsterdam presenting a poster on multi-segment injection with CESI-MS for BNP Proteoform Detection in plasma matrix. This work is an ongoing work with Jennifer Van Eyck's laboratory at the Barbara Streisand Women's Heart Center at the Heart Institute at the Cedars-Sinai Medical Center in Los Angeles. So the focus of this work is BNP. So BNP is a molecule that is secreted in the cardiac myocytes, and it is an important heart failure biomarker because there is a significant correlation between concentration of BNP in plasma and heart failure. BNP is cleaved in small fragments called the BNP proteoforms due to the presence of different enzymes present in plasma, and this degradation occurs rather quickly. So the big question is, because these smaller fragments are really not seen very well by immunoassays like ELISA, the big question we have is how can we reliably use BNP as a biomarker if ELISA is not picking it up? So we would like to have a fast and sensitive method to detect BNP or its proteoforms implicated in the state of heart disease. So for this we used the CSI and with a neutral coated capillary, and we used the high resolution, high mass accuracy mass spectrometry to be able to detect these smaller fragments. But we started, and because we need to monitor at the bedside with the patient plasma to monitor the state of the heart condition, so that implies that we need to analyze multiple data points, multiple time points in this study. So we said, okay, we should then try and use multi-segment injections to increase throughput of this complex analysis. And multi-segment injection, it's a quite simple process. You inject a sample, you have a spacer, and then sample, and then spacer, so on and so forth. And then electrophoretically, you drive this entire train of injected sample plugs across the capillary until detection on the mass spectrometer. In order to accomplish this, we started with a UV detection with a surrogate molecule, just as basic as BNP is. BNP has a PI of just above 12, and the proteoforms between 12.3 to 11.9. So they are all very, very basic. And we used a small PI marker 10 to define initial conditions so that we could apply to downstream to the mass path. So this is the reproducibility we got on a simple experiment with UV detection. This is an example of the method that we used for the multi-segment injection with using optical detection. And then we translated this method into the mass spec, and so we interrogated the spacer length. So we tried different lengths, and the goal here is to be sure that we have very good resolution between different injection plugs, and yet we are able to quantitate everything really well. So we from this experiment, we defined 5 psi 60 seconds as the ideal spacer length. And then we moved on to type of sample introduction. So we investigated both hydrodynamic and electrokinetic injections. And the comparison between the two, based in function, as a function of BNP concentration and peak area, both are very, very similar, but the reproducibility intra and inter-run CV comparison, you can see that hydrodynamic injection also does outperform electrokinetic. However, we do like electrokinetic better because of its selectivity. And so once we move this method from an ideal condition to actual patient samples, we like where the viscosity may vary. Electrokinetic injection may be a better option because we selectively will be injecting the proteoforms of BNP and segregate against other matrices interferences. So this is the level of reproducibility we had with BNP using electrokinetic injections. And inter-run reproducibility were better than 1% overall, which is really good and really important for automated data analysis. And at the end here is an example of the dynamic BNP cleavage in plasma with multiple sample analysis. So each one of these points are 20 time points. And these are the different BNP proteoforms. And what this graph basically shows is the overall formation of each one of these proteoforms over time. And it's not really an enzyme kinetics experiment because we have different enzymes, different substrates, and this is the overall result of breakdown of BNP in plasma. The starting point 42 minutes all the way to 48 minutes. So we have 20 time points down to 840 minutes. So concluding, this method proved to be really good because it does make a very strong case that indeed we can detect BNP in its smaller proteoforms in plasma, so make a very strong case that yes, we can use CESI-MS to analyze this important biomarker. And we are very happy with it. Thank you.
Thank you so much. It was very